Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
Begin writing. The 2022 Nobel Prize in Physiology or Medicine recognized Svante Pääbo's groundbreaking contributions to our understanding of human evolution. This essay undertakes a critical evaluation of Pääbo's Nobel lecture concerning the genomes of extinct hominins and human evolution, scrutinizing its claims regarding the genetic history of Neanderthals, Denisovans, and the implications for modern human origins and adaptations. Our exploration seeks to dissect the specific arguments presented within the lecture, considering the methodologies employed and the conclusions drawn, while also acknowledging the broader scientific context. The following analysis is conducted in a deliberate manner, aiming to fully capture the nuances of each individual claim. Section 1: Introduction to Paleogenomics and the Neanderthal Genome Project. Pääbo's lecture initiates its narrative with a compelling recounting of the nascent stages of paleogenomics, directly addressing the formidable technical hurdles associated with extracting and analyzing degraded DNA from ancient remains. A core assertion emerges: the seemingly insurmountable contamination issues, particularly those arising from bacterial and modern human DNA, presented an existential threat to the accurate reconstruction of archaic hominin genomes. Pääbo credits meticulous laboratory protocols and, crucially, the development of novel techniques for DNA extraction and sequencing as pivotal in overcoming these challenges. He emphasizes the significance of working in a physically isolated, ultra-clean laboratory environment to minimize contamination risks. The development of polymerase chain reaction, or PCR, techniques proved essential for amplifying traces of ancient DNA to detectable levels before sequencing. The lecture places specific importance on the challenges in differentiating authentic ancient hominin sequences from modern, human contamination. The introduction of specific genetic markers, like mitochondrial DNA sequences, allowed for the tracking of each molecule's origin, and this was a crucial milestone. He details the painstaking process of obtaining DNA from Neanderthal bones found in the Feldhofer cave in Germany, highlighting the low yield and fragile nature of the genetic material. A central claim concerns the successful generation of the first draft Neanderthal genome sequence, achieved around 2010, which provided the first comprehensive glimpse into the genetic makeup of our closest extinct relatives. Pääbo posits that this accomplishment served as a catalyst, establishing paleogenomics as a credible and influential field of scientific inquiry, capable of yielding profound insights into human evolutionary history. Section 2: The discovery of Denisovans and their genetic relationship to modern humans. The lecture then shifts its attention to the extraordinary discovery of a previously unknown hominin group, the Denisovans, identified through the analysis of finger bone fragment discovered in the Denisova cave in Siberia. Here, Pääbo makes the claim that the exceptionally well-preserved DNA from this finger bone presented an unprecedented opportunity for genomic analysis. Indeed, the initial analysis of the Denisovan mitochondrial DNA revealed the sequence distinct from both modern humans and Neanderthals, suggesting a separate evolutionary lineage. The lecture highlights the surprising finding that Denisovans, despite their limited fossil record, contributed significantly to the gene pool of certain modern human populations, particularly those in Oceania. Pääbo elaborates on the observation that present-day Melanesians possess a substantial proportion of Denisovan DNA, indicating past interbreeding events. He further posits that this admixture may have conferred adaptive advantages, such as improved immune responses to local pathogens. The lecture underscores the implications of this discovery, suggesting that human evolution was not a simple linear progression, but a complex tapestry of interbreeding and gene flow between different hominin groups. Section 3: Introgression and its adaptive significance in modern, human populations. A significant portion of Pääbo's lecture centers on the phenomenon of introgression, where genetic material from extinct hominins has been incorporated into the genomes of modern humans, and the potential adaptive consequences of this genomic exchange. Pääbo argues that genes inherited from Neanderthals and Denisovans have played a crucial role in shaping the adaptation of modern humans to diverse environments. He cites the example of the EPAS1 gene, a Denisovan-derived variant found at high frequency in Tibetan populations, which confers an advantage at high altitudes by regulating oxygen levels in the blood. Pääbo directly asserts that this is a clear case of adaptive introgression, where a gene variant acquired from an extinct hominin has been favoured by natural selection in a specific environment. He also suggests that other Neanderthal-derived genes may have contributed to traits such as skin pigmentation, immune function, and metabolic pathways in modern humans. The lecture stresses the complex interplay between archaic and modern human genes, highlighting the potential for both beneficial and detrimental effects. Some Neanderthal variants, Pääbo notes, have been associated with increased risk of certain diseases, such as type 2 diabetes and Crohn's disease. Pääbo emphasizes that understanding the functional consequences of introgression is a crucial step in deciphering the evolutionary history of modern humans and our susceptibility to diseases. Section 4. A reconstruction of Neanderthal culture and behavior through genetic information. Beyond the physiological adaptations conferred by archaic introgression, Pääbo explores the potential for genomic information to shed light on Neanderthal culture and behavior. He advances the claim that specific genetic differences between modern humans and Neanderthals may underlie cognitive or behavioral traits that distinguish the two species. The lecture focuses on genes expressed in the brain, highlighting those that exhibit significant differences in sequence or expression patterns between modern humans and Neanderthals. Pääbo acknowledges the challenges of inferring behavioral traits from genetic data, cautioning against simplistic interpretations. He recognizes that the relationship between genes and behavior is complex and often indirect, influenced by environmental factors and gene interactions. Furthermore, he explicitly acknowledges the ethical considerations associated with attempting to reconstruct the cognitive abilities or social behaviors of extinct hominins based solely on genetic data. The lecture emphasizes the need for careful and nuanced analyses, integrating genetic information with archaeological and paleontological evidence to gain a more comprehensive understanding of Neanderthal culture and behavior. Section 5. Challenges and future directions in paleogenomics. Acknowledging the progress made in paleogenomics, Pääbo openly discusses the remaining challenges and outlines potential directions for future research. He identifies the limitations imposed by the degradation of ancient DNA and the scarcity of well-preserved hominin remains. His central assertion is that improved DNA extraction and sequencing technologies are essential for pushing the boundaries of paleogenomic research. He advocates for expanding the geographic scope of ancient DNA studies, emphasizing the importance of sampling hominin remains from diverse regions of the world to capture the full range of human genetic diversity. Pääbo highlights the potential of analyzing ancient proteins, known as paleoproteomics, as complementary approach to ancient DNA analysis. Proteins are often more stable than DNA and can provide valuable information about the genetic makeup of extinct organisms, even when DNA is too degraded to be analyzed. Lastly, the lecture addresses the ethical implications of paleogenomic research, emphasizing the responsibility of scientists to engage with indigenous communities and ensure that their concerns are addressed in ancient DNAs. Section 6. Comparative genomics and the search for uniquely human traits. Pääbo delves into the realm of comparative genomics, specifically emphasizing the search for genetic features uniquely present in modern humans but absent in Neanderthals and Denisovans. Through this approach, he proposes we can pinpoint the genetic underpinnings of traits that define modern human uniqueness. The crux of his argument centers on the identification of genes displaying substantial sequence, divergence, or altered regulation in modern humans compared to archaic relatives. Pääbo concedes the interpretation of these genetic differences requires caution. Many of these changes are likely to be subtle and their functional consequences difficult to discern. He mentions the importance of in vitro experiments and animal models to assess the impact of these genetic variations on cellular processes and organismal development. Furthermore, Pääbo stresses the significance of considering gene-environment interactions in shaping human traits. He posits that uniquely human traits may not be solely determined by genes, but rather arise from the interplay between genetic predispositions and environmental influences. Section 7. The role of RNA analysis in understanding gene regulation in extinct hominins. Pääbo then presents the revolutionary possibility of retrieving and analyzing ancient ribonucleic acid, or RNA, from extinct hominin remains. The core claim is that ancient RNA, while even more fragile than ancient DNA, can provide direct insights into gene expression patterns in extinct hominins. Pääbo recognizes the extreme technical challenges involved in recovering and analyzing ancient RNA, given its rapid degradation after death. However, he suggests that under exceptional preservation conditions, such as those found in permafrost environments, it is conceivable that fragments of ancient RNA could be recovered. He elaborates that by comparing RNA expression patterns between modern humans, Neanderthals, and Denisovans, scientists could potentially identify genes that were differentially regulated in these groups, thereby gaining insights into the functional differences between them. Pääbo emphasizes that the field of ancient RNA research is still in its infancy. It holds immense promise for advancing our understanding of gene regulation and evolutionary. Section 8. Epigenetics and its potential contribution to the understanding of hominin evolution. The lecture pivots to the field of epigenetics, exploring the potential role of epigenetic modifications in shaping hominin evolution and phenotypic differences. Pääbo posits that epigenetic marks, such as DNA methylation and histone modifications, can alter gene expression without changing the underlying DNA sequence. Furthermore, these epigenetic changes can be influenced by environmental factors and potentially be inherited across generations. Pääbo suggests that by studying epigenetic patterns in ancient hominin remains, scientists could potentially gain insights into how environmental factors influenced gene expression and phenotypic development in our extinct relatives. He acknowledges the technical challenges of studying epigenetic modifications in ancient samples, given the degradation of epigenetic marks over time, but Pääbo maintains that advancements in analytical techniques may eventually allow for the reconstruction of epigenetic landscapes in extinct hominins, providing a more holistic view of their biology and adaptation. Section 9. The ethical considerations and broader implications of paleogenomic research. Pääbo broaches the crucial topic of ethical considerations associated with paleogenomic research, particularly concerning the handling of sensitive genetic information and the potential implications for societal perceptions of human diversity. He underscores the importance of engaging with indigenous communities and respecting their cultural heritage in ancient DNA studies. Pääbo emphasizes that the genetic information obtained from ancient hominin remains should be used responsibly and ethically, avoiding any interpretations that could perpetuate harmful stereotypes or discriminatory practices. He advocates for transparency in research practices and open communication with the public about the findings and implications of paleogenomic research. Pääbo further argues that paleogenomic research can contribute to a deeper understanding of human history and diversity, promoting empathy and understanding among people from different backgrounds. He explicitly cautions that we should strive for a world in which the unique history of every population is understood and respected, and which this knowledge contributes to a more equitable world. For Section 10 conclusion, Pääbo's vision for a future of human evolution studies. In summation, Pääbo's lecture articulates a profound vision for the continued exploration of human origins through paleogenomics. He concludes by reiterating the transformative impact of ancient DNA research, stressing its capacity to illuminate the complex interactions between modern humans and no-extinct hominin groups. A central tenet of his final remarks centers on the promise of technological innovation, heralding the potential of refining DNA extraction, sequencing methodologies, and computational analyses to provide even greater resolution into both the genomes and the behavior of extinct hominins. He champions the continued integration of paleogenomic data with archaeological, paleontological, and anthropological evidence to provide the most holistic understanding of human evolution. Pääbo ultimately offers the insight that scientific progress is at its best when it promotes a deeper appreciation of our shared human heritage, fostering empathy and respect across cultures. His lecture stands as a testament to the power of scientific inquiry to unravel the mysteries of our past and inform our understanding of the present. The meticulous analysis of archaic hominin genomes, as championed by Pääbo, has indelibly reshaped our comprehension of human history, revealing a narrative far more intricate and nuanced than previously conceived. His contributions will undoubtedly continue to inspire and guide future generations of researchers as the field of paleogenomics continues to evolve.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.